Okay, yeah. So as I was saying, today we'll look at kind of more of the physiology side of all the little proteins inside of muscle and how muscle contraction works. And so the general theory for how muscle contraction works is called the sliding filament theory. So we'll spend most of the class on that, and then we'll just at the very end kind of look at the muscles involved in taking a step. Okay, so it'll be kind of a transition over to that. So here are the muscles, or the filaments inside of muscle that we talked about last time. Okay, so kind of our key players here would be the myosin. Okay, the myosin is the one that has the heads. Okay, that's gonna be a major part of the action. Okay, so it grabs onto actin and it's going to pull actin. Okay, those myosin is known as the heavy chain. Okay, and then we've got the light chain protein, which is called actin. Actin is the one that kind of gets grabbed onto by the myosin, and it's the one that slides. Okay, so it's going to get pulled on by that myosin, and it's the one that slide in the sliding filament theory. Okay, tropomyosin we talked about last time as well. So this is another light chain. This is the one that's covering the actin-myosin binding site, so it's kind of like blocking it. It's getting in the way. Troponin we talked about is the one that calcium binds to. That its shape is going to change when calcium binds to it, which then is going to help us to allow for that cross bridge to form between actin and myosin. Calcium's key in binding to troponin and changing its shape. And then ATP, or adenosine triphosphate, is key in not actually causing the sliding, but cocking the myosin head, putting the myosin head into its power packed position. So those are kind of our key players here. And we're going to dive into two separate processes. But take a second and with this little question over here on the side of the slide, decide what you think with that GIF there. So which one above is tropomyosin? Check with the people around you on that and send it in on your clickers when you're ready. Not hard to see over here. The podium's in the way. Yeah.
I guarantee you it's in my car. Oh no, here.
You got it.
Oh, wait. Or, yeah.
Right.
All right. So all right. I think there's more of you need to send it in. I only see 36, so send it in. Are we struggling with the actual color itself? They are kind of weird colors. There we go. I think we almost got everybody got 46. Show you what you guys got here. So yeah, so we're kind of between B and D there. With those two. Yeah, so go ahead. Yeah, I know that's kind of what I wondered. But yeah, I was calling purple the one that's right here. This little guy. Yeah, yeah. And then this longer one would be the blue one. Yeah. Okay. Maybe you guys were thinking that this was purple, the long one that I'm calling blue. Yeah. Okay. All right. I know they're kind of weird colors. So the tropomyosin would be this blue one here that I'm calling blue. It's really hard to try to mark on it because it keeps moving. But we'll have another image that makes it clear. Let's just go to this image right here in the notes. This one is clearly blue, right? Yeah, it's kind of like this one here, right? It's the one that goes along the whole thing. So we can see that here. It's this one that's going along the whole thing here, right? Not the little chunk in the middle, but the little more like rope part between those. Got that? Make sense? Okay. All right. So what we're going to talk about is these two separate processes that are both part of the sliding filament theory. The first one is called excitation contraction coupling. And so this starts with what we call an action potential. This is the language of the nervous system, right? So this is when an electrical signal gets sent down a neuron, and that signal gets passed over to the muscle cell, and then we activate the muscle cell. Okay, so that's kind of where we're starting, and then this ends with calcium binding to troponin. So it's kind of like the preparation for contraction, right? So we're thinking about this one as the prep for contraction. Okay, and then that opens up the cross bridge opportunity, possibility. And so then we move into cross bridge cycling. So this is where we're actually going to have the shortening of the muscle cells and shortening of those sarcomeres. Okay, so we'll start with excitation contraction coupling. And we'll start with it by watching this video. This, I've kind of have the steps listed out here. And so what I'd recommend doing while we're watching the video is just kind of try to follow along with these steps, kind of try to figure out where they connect in the video. And then when the video is done, we will kind of, you'll go through and you'll fill in what's happening here each step of the way. Okay, so let's go ahead and play that video. And here we go.
Muscle contraction is at the basis of all skeletal movements. Skeletal muscles are composed of muscle fibers, which in turn are made of repetitive functional units called sarcomeres. Each sarcomere contains many parallel overlapping thin, actin, and thick myosin filaments. The muscle contracts when these filaments slide past each other, resulting in a shortening of the sarcomere and thus the muscle. This is known as the sliding filament theory. Cross-bridge cycling forms the molecular basis for this sliding movement. Muscle contraction is initiated when muscle fibers are stimulated by a nerve impulse and calcium ions are released. The troponin units on the actin myofilaments are bound by calcium ions. The binding displaces tropomyosin along the myofilaments, which in turn exposes the myosin binding sites. At this stage, the head of each myosin unit is bound to an ADP and a phosphate molecule remaining from the previous muscular contraction. The myosin heads release these phosphates and bind to the actin myofilaments via the newly exposed myosin binding sites. The two myofilaments glide past one another, propelled by a head-first movement of the myosin units, powered by the chemical energy stored in their heads. As the units move, they release the ADP molecules bound to their heads. The gliding motion is halted when ATP molecules bind to the myosin heads, thus severing the bonds between myosin and actin. The ATP molecules are now decomposed into ADP and phosphate, with the energy released by this reaction stored in the myosin heads, ready to be used in the next cycle of movement. The myosin heads resume their starting positions along the actin myofilament and can now begin a new sequence of actin binding. The presence of further calcium ions will trigger a new cycle.
Okay. So, yeah, so let's take a look at this. That kind of focused more on the cross bridge part. It talked a little bit about the excitation contraction coupling too, but let's just kind of walk through a couple of these steps first, and then I'll let you go ahead and do the fill-in part. So, again, the key starting point is when the motor neuron, which is a neuron coming from the nervous system, from the spinal cord, going out to the muscle, it sends a signal, which that signal is called an action potential to the muscle. Okay, that signal is going to arrive at the neuromuscular junction. So that's that space between the nerve and the muscle. And then there's a little space in between that whole part. It's called the neuromuscular junction. Okay, the key there is that we're going to start another action potential on the muscle now. So we started with an action potential or a nerve signal on the neuron. and then we pretty much transferred that to the muscle cell. Okay, that action potential is going to spread along the muscle cell membrane and those T tubules. Remember the T tubules that helped us get that nerve signal deep down into the muscle cells. Okay, that activates that sarcoplasmic reticulum, which is what stores calcium for us, right? Calcium is released, calcium binds to troponin, makes troponin change shape, and then that moves tropomyosin, right? So now we have this open myosin binding site on actin. Okay, so with that, take a few minutes with the people around you and just fill in, kind of write in what you see going on with each step here. Okay, so there's kind of 4 slides here. Yeah, so go ahead and start with that.
See the motor neuron setting it.
So, yes.
Are you also gonna give us stuff to fill in or do we?
Oh, yeah, there's nothing that uncovers anything, so, yep, it's just you.
I just didn't wanna write where there's gonna be stuff. to-
Yeah, yeah,
Okay, oh.
right on, it. Yeah, totally.
But then with more, this is probably step 3 right here. This is step 2. There. Wait, this is mostly someone's. The next I see after the same thing, steps 1 or 3. So would it be step this is or that should be there's 3 steps. 1, 2, 3 for the section. Oh, then we did. I think for the first year, I was going to say, I don't
Like 1 or 2. Of that info. Writing a card, but that would be this one.
It's just the first step. To see like those stuff. That's so when the nerve sends a signal. So then 9 would be this, right? Or. That's fine. And then. I think this was. And then basically turns on every. Yeah, I was going to show you like. That's yeah. I don't know.
All right, let's go ahead and come back together here. Okay, so we'll kind of walk through what's going on here. Okay, which step numbers did you think are on this image, this first image right here? Obviously 1, right? 1 is, what does it say? Motor neuron sends a signal, right? So we're getting the signal coming down the motor neuron, right? So it's step 1. Step 2 was arrives at the neuromuscular junction. We're seeing that, right? So this is the neuromuscular junction. NMJ. So that includes part of the neuron, the space between, and part of the muscle cell. Remember, we have this specialized part of the muscle cell membrane. What was that called? The motor end plate. Remember the motor end plate that is all folded? There's a bunch of folds in it, so we can put a bunch of these receptors for the neurotransmitter there. We'll come back to the neuromuscular junction later, and we'll kind of fill in some of the details there with sodium and stuff. We're not doing that yet. So you just are responsible for the kind of stuff on this slide here. As far as there's some extra details there with sodium and stuff that we're not getting into yet. Yeah, so it arrives at the neuromuscular junction, and then what's the third one? It starts an action potential there, right? So we see that action potential. It was started on the muscle cell membrane, so now it's traveling down the muscle cell membrane, and we're kind of just starting to see a little bit of the T-tubule, but we'll see that more on the next slide, really, right? So this is really, yeah, steps 1 through 3, right, would be the ones from that previous image. Okay, questions on that? Okay, so then we go to the next step, which we're seeing the action potential continuing. That was this green line, right? And now we see the T-tubule, right? So it's going to dive down the T-tubule so we can get deeper into the muscle cell, right? And so with that is really just step 4, right? That was step 4. And then we go to this one where we can see these blue structures. What is that blue structure? It's not labeled there, but what is it? Hmm? There we go. Adriana. There we go. Thank you. Sarcoplasmic reticulum. All right, so that's that guy. It stores calcium for us. You can see that it's actually connected to the T-tubules. All right, there's actually a protein that connects those two structures. And so when the action potential goes down the membrane here, it changes the shape of this protein, which then changes the shape of that other protein that it's connected to, which is the calcium channel too. So then calcium is able to come out, right? So we're releasing calcium from the sarcoplasmic reticulum. So that's step 5 there, right? So we're seeing that. Okay. Questions on that so far? Okay, and then our last image here contains the rest of the steps. So we see calcium came out, right? It gets dumped into this space behind all of this stuff, which is where the actin and the myosin and the troponin and all that stuff is, right? In the sarcomere. Okay, so we can see before calcium binds, right? So this is at rest, right? And then this is after calcium is released and binds to the troponin, okay? So let's take a second and just remember what we talked about early on with protein structures, right? What was that first level when we just string a bunch of amino acids together? What do we call that level of protein structure? Primary, okay, and then we went to secondary, tertiary, quaternary, right? That first level is all really strong covalent bonds, peptide bonds. So those don't really change much. But once we get beyond that to the secondary, tertiary, quaternary, we're relying more on hydrogen bonds, relying more on the interactions with the water, relying more on things like if a positively charged amino acid is next to another positively charged amino acid, they're going to push away from each other, right? All of that gives proteins their shape. And so when we take this troponin protein, it has a binding site for an ion that has a positive 2 charge to it, right? So there must be some amino acid there that has a binding site for that. And that amino acid probably has a negative charge on that part, right, on its R group. And so when the calcium comes and binds to it, it's no longer negatively charged. That could have neutralized it, or it could have made it positively charged. And so now the way that it interacts with the amino acid next to it is going to be different, right? So maybe there were 2 positive ones that pushed away from each other. Or let's say there was 1 positive, 1 negative, right? And those went towards each other. Then we get this calcium that comes and binds to it. That one's no longer negative. It's positive now. So now it pushes away instead, right? That's what it means by changing the shape of troponin, right? We're messing with the structure of that protein, right? And it just moves it a little bit, right? And it's also attached to this tropomyosin one. And the tropomyosin is really the one that's kind of covering up all of those sites where the myosin head is going to bind, right? And so if you just think about that for a second, this is a mechanism that allows pretty much all life forms that move to move, right? This is what leads to resting muscle, this right here, right? And this is what allows muscle to contract. When we just change the shape of this protein and it moves, it also involves the myosin head, which we'll talk about in a second too. But that one is the same thing. When a phosphate binds to the myosin head, that's what changes its shape, right? And all of this is just because we have all these little amino acids in the exact perfect position that gives them little pockets where certain things combine to them, then it changes their shape, right? And so it's this incredible little structures that are just affected by these tiny little ions, right? Or tiny little molecules, which is what animates all of biology, right? This mechanism is what allows all animals to move, even plants that can move a little bit use a similar mechanism to this. So it's really this universal thing that allows for movement across life. So this is all just kind of preparing us for contraction. And so we end here with the binding site open, right? So that's where we end. What steps are those? Those would be, I guess, 7 through 9, right? Yeah. Pretty much those last steps, 7 through 9 there. And with that, no, we skipped 6. Huh? Where was 6? 6 was calcium is released. Yeah. We're seeing calcium release here, so this is 5 and 6. Yeah. Okay. Obviously, we're seeing calcium coming out there too, so it's both. But okay. Questions on that? Okay, so that's all the preparation for contraction, right? So now we got the binding site open. Okay, so then our next process is cross-bridge cycling, and this is actually where muscle shortening occurs, right? There's really just kind of 4 primary steps to it, and it's a cycle. So you can kind of start in different places. We've just kind of chosen to start here. This was in the video too. Okay, so we're going to start with the cocking of the myosin head. Okay, and one analogy that I think is kind of helpful for this is if you've ever shot a gun before, a pistol, or probably most of you haven't, but the way that you do it is you take your thumb and you cock the hammer back, right? But that's not when the bullet comes out, right? That's just cocking it, right? So that's why this is called cocking of the myosin head, is because with a gun, you put your energy into it before the bullet comes out, right? It's the same idea with this. We're using the energy from ATP before the muscle actually shortens, right? So we're cocking the myosin head, we're putting it into its power pack position with the use of chemical energy, which is ATP, right? But the muscle's not shortening yet. Okay, so ATP hydrolysis is the way that we break down ATP. Remember, ATP has those 3 phosphate groups on it, right? The bonds between those phosphate groups are considered high energy bonds, meaning that they're like, those phosphates are like ready to bind to something else, right? And when they do that, that phosphate changes the shape of whatever it binds to. And so that's kind of the putting its energy on. And so when we do hydrolysis of ATP, we're breaking off on those ATPs, and that creates some water too. And that's what cocks the myosin head. Okay, so that's step number 1 is ATP hydrolysis, which is going to cock the myosin head. Now that it's cocked, the myosin head can attach to actin, so then it goes up and attaches to actin. And then we finally get the shortening part. That's called the power stroke. The power stroke is really when the ADP and phosphate come off, then it goes back to its original position, but it's attached to actin when it does that. So it's already attached to actin, so it just kind of pulls actin with it when it springs back. And that's really the shortening. Okay. And so from there, then the myosin can detach from the actin, but only when a new ATP comes. So a new ATP molecule comes, it gets broken down. That's what allows the myosin head to come off. Okay. So for when you guys looked at the donors in lab, the cadavers, right? Their joints are super stiff, right? You can't really move their joints around much. It's because all of their muscles are in rigor mortis. And rigor mortis is caused by not having any new ATP, right? Because if you don't have any new ATP, you can't get the myosin heads off of actin because that's what pulls it off, right? So right after the power stroke, myosin detaches from actin, but only when there's a new ATP to help it come off, right? So when somebody dies, they're not breathing in oxygen anymore, right? So they're not producing any more ATP. So their muscles are all locked up together, right? And that's rigor mortis. Okay. So let's take a sec and do what we just did, but through these steps now. So we've got 4 images here. And then there's one here that puts them all together. Okay. And I want you to answer this one on your clickers. Okay. So which one is step 1 from the previous slides here? But go ahead and fill in all of this, what's happening with each one. And then you'll answer this question here on your clickers. Okay. Go ahead.
This is the last.
So is excitation, contraction, coupling, is that just the preparation and then bridge cycling is the active?
Action Jordan. Yeah.
Okay.
You got it.
Got it.
Mhm. Yeah. Question?
So if it's, they need ATP to like release it. But so that's step 1, would it be D? Cause it's clocking.
That's a great thing to talk about with your teammates here. You'll probably learn something along the way by dissecting that together.
I think B is.
No, because that would be the ATP. Yeah. There's new ATP. This one I have. That's one. So ATP is now. So I think it's C. Step 4 is
Oh, wait, at least.
This one's
Hey, you know, quick section class after class? Yeah, yeah.
It's, I forgot we have labs again.
All right, so you're sending in your answer to this question here. Which step do you think is step 1 from the previous set of slides? All right, there's about 7 or so more of you to send it in, looks like. All right, I'll leave it open for a sec so you can continue sending that in if you haven't yet, but let's start here. Okay, so in this set here, we're starting with the cocking of the myosin head, right? So this is that cocking phase, right? So it's going from being ATP to being ADP and phosphate, right? So we're breaking down the ATP in this step here. And when phosphate binds to this myosin head, it's just like the calcium and troponin, it changes the shape of this protein, right? It changes it in a big way. It moves it quite a bit. And that's the cocking of the myosin head. Okay, so that's us putting our energy into it. That's like pulling the hammer back on the pistol. but not pulling the trigger yet, right? We're just pulling it back. So we're kind of loading up the energy in it. So it kind of has potential energy now, right? So that's the first step. Okay. Second step is the myosin head goes up and attaches to actin. Okay. So it's already cocked. It's already in its power pack position, but now it attaches. Okay. So we got that attachment. Third is we see ADP and phosphate come off of the myosin head and it's attached to actin and it just is stuck on there. And so it takes actin with it, right? So this is the sliding, right? This is when we're seeing the sliding. This is muscle shortening. Right, so this is when the muscle actually shortens. Okay, and then we move into the last step, which would just be that new ATP comes, right? Which is what allows for the myosin head to be detached from actin. Okay, and then we could immediately go right back to step one, right? Where that ATP turns into ADP, attaches again, right? So that's the whole cycle there. Okay, so then when we look at it in this form, it looks like about 80% of you are saying C is the same one from step one in the previous slides. So step one is kind of, it's a bit of a mix of these two, right? But it is mostly this one because we're seeing that the ATP is broken down and we're detaching, right? But it's a little bit different, right? Because but we do see that it's cocking. That's the key part that's the same here, right? Is we're going from this position, which is a smaller angle, right? To this one where we're cocking the myosin head. So that is the key part of that one. So cocking phase right there. So that's where we're like using the energy from ATP. So yeah, C is the correct answer there. But we're not seeing it as ATP like we did in step one here. But besides that, it's pretty much the same. That makes sense? Okay, so then so same thing, right? We cock the myosin head, it attaches here, ADP and phosphate come off, we get the sliding, get the shortening, okay, the new ATP comes, it detaches, and then we go back to cocking it again. That's the cycle. Okay. Questions about any of that? Looks like you want to ask a question, Matthew. Yeah, like the angle is different. Yeah. 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 I don't know. It's probably somewhere in the middle of the two. It is unique to see when it gets this straight. I've never seen an image created like this that shows up quite that straight. It's probably more like this, but then this is like a bit extreme too compared to some of the. I've seen some papers where it actually shows the protein structure in itself, and so it's probably somewhere in the middle of the two. But yeah, good question. Other questions? Okay, so if we're just going to put those two things together, right, we're going to start with excitation contraction coupling. We don't need to know about acetylcholine yet. We'll get there. But we're sending that action potential down, right? Action potential started on the muscle cell, down the T tubules. We get calcium release, binds the troponin, do that whole cycle we just talked about. And that's kind of the big picture. We'll go back soon and we'll talk about more details of what happens here in the neuromuscular junction. And then when we get to the nervous system, we'll go back another step and talk all about how action potentials occur and all of that. So we'll kind of keep building on this. But for now, this is where we're at. So any questions on that? Okay, so a bit of a transition to kind of just a kind of plugging in how which muscles work to work while we're taking a step. Okay, and so we're going to think about this in terms of both our weight bearing leg and our leg that we're using to kind of push off and then swing forward. Okay. It's always hard in a classroom like this where you guys can't see my feet. So I want to just walk up here so you can see me, but that's not going to work so well probably. But so the idea here is we're going to be taking a step with your right foot. Okay, so it's showing this. So I'm like taking a step with my right foot. That means my left foot or my left leg is the weight bearing leg. Okay, so we'll start with that one. That one's pretty simple. The key is when we lift our right foot off the ground, we have to activate some muscles in our hip to keep our pelvis parallel with the ground, right? If we didn't activate anything when we took our right leg off the ground, our hip would fall like that, right? So any guesses on which muscles might be contracting over here to stabilize our pelvis so we don't fall down like that? Some glutes, yeah, for sure. And in particular, gluteus medius. Because that's our big one that does abduction, abduction, right? Which would be like this motion with our hip, right? And so if the foot's not allowed to move, it can contract and just hold us in place, right? So it's acting as a stabilizer muscle instead of a mover, right? But it's doing that, it's just holding our pelvis in place, okay? So that one's easy. Okay, so that's my left foot if I'm taking a step with my right foot, okay? Now let's look at the other one. Okay, so this is the leg that's actually going to be moving forward, okay? So we've got kind of we're breaking this up into two different phases. The first phase is when we push off. Okay, so when we push off, so I have my right foot backwards. Okay, I'm going to push off. First of all, I'm going to do plantar flexion with my ankle, right? Which muscles help us with plantar flexion? Our calf muscles, right? So gastrocnemius, soleus. Okay, so gastrocnemius and soleus are big ones there. Okay, so we're pushing off. We're kind of pointing our toes, right? In that motion, we're also extending our hip a little bit, right? It's not really what we think about when we do that, but our hip does go back a little bit when we push straight out. What would be some of our hip extensor muscles? Hamstrings, glutes again as well, right? So what would be the names of those hamstrings? What's the one on the lateral side? Biceps femoris, and then we got my tendinosus and my membranosus, okay? So we can name those, okay? And then this one's kind of weird. The knee part of this is kind of strange, but when you take a step, there's a little bit of knee flexion that happens for a second. It's kind of like loading up your knee before you push forward, okay? So with a little bit of slight knee flexion, what are our knee flexor muscles? Hamstrings again, right? And gastrocnemius crosses over the knee as well. And so it acts as a little bit of a knee flexor. Okay. So I kind of helped us on that one. What I want you guys to do for a sec is stand up with the people on your team, with your partner, whoever next to you, and kind of try to do that. So kind of try to do the push off thing, kind of feel how you might have those motions, and then do the swinging forward part too, because it tells you what motions it's going to happen on the swinging forward part. You kind of think about which muscles you would plug in there. Try that for just a sec, and then we'll come back together. You can take a lap around the classroom if you want. Take some steps.
Or that just being a So it might just be quiet. I'm thinking I have.
So it's not, and the hip extension is not.
You could do glutes.
It also is.
Yeah, you could.
And.
It's not as much. The glutes are more active when you're up
Okay.
in flexion more, like when you're going upstairs. That's really when you get your glutes. But that's why I didn't write them there. Quads for what?
In hip extension.
Quads don't have any hip extension action. There's one quad that's a hip flexor.
Yeah,
Right?
yeah.
Yeah, you can fill that one in down there.
Okay.
Yeah.
Correct.
The knee part's kind of weird for sure, because the knee kind of does multiple things.
Wait,
but-
which part of that is that?
Yeah, so it's like you kind of like, you push off, and so like when it bends down like that a little bit, see how it kind of like, yeah, like it kind of flexes a tiny bit before you push, so that's kind of what it is, but it's a slight little motion. Yeah.
Have lunch.
I don't know.
All right, let's go ahead and come back together here. Okay. So yeah, so we did our push off part. So we got plantar flexion in the ankle. We got hip extension in the hip, right? We got flexion in the knee. Okay. And if there was a question like this and you added some more hip extensors like gluteus maximus, that would be fine. The gluteus maximus is more active when you're in hip flexion already, like when you're stepping up and then pushing up. When your hips kind of already somewhat extended, the hamstrings are more active, but that's not a big deal. You don't need to know that. If you plug a muscle in there that is a hip flexor or a hip extensor in this case, then that's fine. Yeah. Okay. So now we're going to do hip flexion, right? So now we're like swinging our whole leg forward, right? So what's our primary hip flexor muscle? I think that was a question on the TBL day, wasn't it? Iliopsoas, right? Yeah. Okay. Iliopsoas. Okay, but then we've also got that quad muscle that helps us with that, rectus femoris. We've also got that little pectineus, which is a hip flexor as well. Okay, so out of our 4 quads, there's one that starts on the pelvis, right? That's rectus femoris. The rest of them start on the femur. Okay, so the one that starts on the pelvis is a hip flexor as well. Okay, and then when we swing our leg out, we're going to kind of like kick it forward, right? We're going to straighten our knee out when we do that. So knee extensors, what are those? Quads, okay. Starting with our lateral one, what's that one called? Vastus lateralis. Keep going. Vastus intermedius, vastus medialis, and the one we already said, rectus femoris, right? So yeah, those are the quads. Okay. And then as we kick our foot out, we want to pick our toes up a little bit, right? If we don't, we might trip on them for one thing. But we also want to land on our heel when we land, right? So we dorsiflex our ankle. Okay. What's our big dorsiflexor in the lower leg? Tibialis anterior for sure. Okay. And then we also get some help from the one that extends our toes too. What would that one be? Yeah, so that'd be tibialis anterior extensor digitorum longus. Okay, we're going to have extensor digitorum that we'll see as well, and then we'll also see another one so that's in the forearm, but then in the cat we'll see the forearm one is called extensor digitorum communis. So there's kind of 3 of those different extensor digitorum ones. Try to keep them straight in your head. That's the tricky part, but there's a bunch of them. Okay, so that's the idea. Just kind of understanding what motions happen, filling those in, and then it would start again on the next side. So that's all we got, guys. We'll get done a little early. Have a great rest of your day. See you guys on Wednesday. Yeah.
Recording down. That is